Hello, good afternoon. This is Umma Fatema from Dr. Tomokazu Kawashima lab. Today I will talk to you about my research proposal. It's basically my journey of understanding since January this year when I first started my research on Arabidopsis fertilization with Dr. Kawashima. My researches focus on molecular and cellular dynamics in the Arabidopsis female gamete for fertilization. The very first understanding I had on fertilization on Arabidopsis is the importance of fertilization in flowering plants. Why we should care about fertilization in flowering plants? We should care about fertilization in flowering plants because during fertilization, upon fusion of male and female gametes, we get seeds. These seeds, in one hand, provide the major sources of our food, and on the other hand, it continues its generation throughout time and maintains the continuous supply of food for the human being. Fertilization in flowering plants is different than that of animals. In case of animal fertilization, microtubule pulls the female pronucleus toward the sperm pronucleus. But in case of flowering plant, sperm nucleus migrates to female nucleus. And instead of using microtubule, it uses F-actin for its migration. But how does F-actin regulate sperm nuclear migration? We don't know. Dr. Kawashima has investigated the dynamic F-actin movement in the female gamete cell in Arabidopsis. And he found that there is a constant inward F-actin movement in one of the female gamete cells, which is central cell. When he disrupted the function of this inward F-actin movement, he observed a failure of sperm nuclear migration as well as fertilization in this Arabidopsis central cell. We also don't know much about this inward F-actin movement in the central cell. How does it control? Except the role of a small rho GDPS, which is present on the plasma membrane in its active form and regulate this inward F-actin movement. Dr. Kawashima has also investigated the effect of motor protein myosin in the dynamic F-actin movement in the central cell, because this motor protein is responsible both for organelle motility and F-actin movement in the cell. This myosin protein has binds to organelle with its tail domain and binds to F-actin with its motor domain and act as a carrier for carrying the cellular organelle. When he investigated the F-actin dynamics in the control central cell, he found here the nice inward F-actin movement. He marked the actin with lipid peptide tagged with fluorescence protein venom. But when he investigated the treated sample with myosin inhibitor, he found that the movement of F-actin became stopped in the myosin inhibitor treated sample. Based on this information, my task in the lab is to investigate myosin's role in the inward F-actin movement in the central cell, as well as to identify other factors that may be involved in this inward F-actin movement in the central cell. In my today's talk, I will only talk to you about the myosin's role in the inward F-actin movement in the central cell. To understand the myosin's role in this inward movement, first I analyzed the different classes of myosin across the different kingdom. This is the phylogenetic tree of the myosin superfamily based on their motor domain. There are 17 classes of myosins present across the different kingdoms on the earth. Among these 17 classes, only 2 classes are present in flowering plant Arabidopsis, class 8 and class 11. These 2 classes are different in terms of their configuration and also in terms of their distinct function. Myosin 8 is mainly responsible for cell-to-cell communication and cell division, whereas myosin 11 is mostly responsible for organelle motility and effect in organization. There are 30 members of this myosin family, and there are 4 members in the myosin 8 family. As our interest in effective dynamics in the central cell and sperm nuclear migration during fertilization, we further wanted to investigate the role of myosin class 11 family. But the problem is there are 13 members in this family. It's not possible to work all these 13 members at the same time. Therefore, to narrow down our candidate myosin, we investigated the publicly available transcriptional profiling data. We generated a heat map based on their expression GA score in the female gametes and in different compartments of the seed throughout the developmental period. In this triangle, it represents the developmental period of the seed. The narrowest represents the younger development period, and the wider edge represents the older development period. We also narrow down our screening only on the central cell, as our focus is on the effective dynamics in the central cell. We ranked our all 13 myosin based on their expression on the central cell from 0 to 10 score. 0 means lowest score and 10 means highest score. And we are able to identify 2 candidate myosins which ranked 8 in the central cell expression. Based on this heat map, this XIE is prevalent in pollen and XIG has prevalent expression in chalidial endosperm. We further confirm their expression in the central cell by generating transcriptional line. The native promoter of these candidate myosins were hooked to fluorescence protein, venous. We only got the promoter activity of myosin XIG and found their expression on the central cell and in the synergic cell. But unfortunately, we didn't get any expression of myosin XIE in none of the female gamete cells. We further wanted to have the role of myosin G in effective dynamics. We again investigated the effective dynamics in wild type and in myosin mutant background. And what we found here that in case of wild type, there is a nice inward effective movement in the central cell, which is represented in different color code. 0 means green, 1 minute is represented as magenta, 2 minutes in cyan, 3 minutes in yellow. But in case of myosin mutant, the inward movement becomes disorganized, and instead of going inward toward the center of the cell, the movement becomes outward of the cell. So how does myosin play this inward effective movement? We don't know. Therefore, in our lab, we are investigating the sublocalization of myosin G by generating full-length myosin G fused with fluorescence protein. I will also verify the myosin G sublocalization in the central cell by immunolocalization. I'm also interested to understand the cellular dynamics of myosin G in the central cell by time-lapse live cell imaging movement of XIG in the mature unfertilized central cell, because myosin G has active movement in the central cell, and we hypothesize that this active movement may be responsible for the dynamic effective movement in the central cell. I would like to also investigate the cytoplasmic streaming in myo-G mutant background, because when we disrupted the myo-G gene, we got the movement of the effectin became inhibited. Therefore, we wanted to know whether myo-G has any effect on the organelle movement in the central cell or not. Finally, I would like to identify MyoG interactors by immunoprecipitation, because we also want to know that whether MyoG has any either direct effect or indirect effect in the effective dynamics in the central cell or not. If the effect is indirect, we are expecting to get organelle protein as its interactors. So my work in the lab is filling up the current knowledge gap by providing the answer of how flowering plants utilize effectin for gamete-nuclear migration. It will also provide a foundation for linking environment with molecular factors for sperm nuclear migration. Finally, I would like to give thanks to Dr. Tomokazu Kawashima for giving me the chance to work with him in this nice project. I would like to give thanks all of my lab members, especially Fote, Anthony, and John, for their tremendous support in the lab, and all the audiences for your patience hearing. Thank you.
I was wondering about your immunolocalization. Do you have an antibody specific for that particular myosin?
Yes. Yeah, so currently we are working on a peptide-based antibody against myosin G, and it seems like we are getting nice data using this antibody. Any questions from Princeton? Oh yeah, you. Yeah.
What that you're doing this knowledge is gaining. What techniques have you learned, or do you still need to learn?
Okay, so the very first technique I'm learning the gateway technology, because we generated a lot of transgenic line. As I showed you that, like, we generate transcriptional line, translational line, so it's the gateway technology. Other technique is the live cell imaging, because like mostly our research focused on the live cell movement of effective dynamics in the live cell. And also I'm learning Western blot for immunolocalization to find the interactors of MyoG.
I have a question. What are your mutations in actin have on fertilization?
Yeah, so I have two answers for you. So when we disturb the function of actin by its dominant negative form, then we get a reduction of fertilization, like failure of fertilization. But in my case, when I showed the image of XIG mutant, we didn't get any fertilization defect in our cell. So I'm just assuming that maybe MyoG has some complementary gene in the cell.
All right, thank you very much. Next up, we have Ms. Madison Kramer with her presentation on spray nozzle effect on herbicides.
All right, thank you for that introduction. Again, my name is Madison Kramer, and today I'm going to be talking about the influence of spray nozzle design and weed density on herbicide coverage and deposition. Before we start, I'm going to go through a quick overview. So we're going to talk about introduction, issues regarding dicamba, some restrictions when applying the chemical. We'll get into some drift-reducing agents, and we'll end with some objectives for the introduction. We'll do a methods and materials with my trial design and our two main factors of my project. Then we will do some data collected. We'll talk about those results, and then we'll end with the conclusion and have time for questions at the end. So dicamba injury due to soybean has been a major concern. In the state of Kentucky, we experienced 35 to 45,000 worth of acres that were damaged, but in 2018, we only experienced 6,000 acres. Does this mean we're spraying less dicamba? Absolutely not. It just means more dicamba tolerant soybeans have been planted. So in the state of Kentucky, we have to keep in mind that there are sensitive crops that are being grown next to our soybean fields. And we have to worry about tobacco. In 2018, 500 acres worth of tobacco was damaged. This is huge for the state of Kentucky because tobacco is a very high valuable cash crop, and if those plants are damaged, the farmer could ultimately lose his contract. We also know that the EPA just released or extended that as a chemical label for 2 more years. So it's good. We can still, spray this chemical to control our problematic weeds, but we have to keep in mind those restrictions. We also have to know that many homeowners that have, landscapes, trees, and like gardens, they know what that chemical looks like. So some restrictions when applying, we have wind speed, we have a minimum and a maximum we have to obtain. Boom height can affect our drift potential as well as the pressure. And today I'm going to be talking about those different nozzle types. So there are many nozzles that have been approved to spray dicamba. And what these nozzles do is they increase our droplet size spectrum. So they produce nozzles that vary from very coarse to extremely coarse. Right here, or ultra coarse, right here I have 2 examples. We have our Hypro Ultra Low Jet. They are manufactured by Pentair and they produce a very coarse droplet. And then we have our Turbo T-Jet Induction. They produce an ultra coarse droplet. This is the biggest droplet size we can get. These 2 approved nozzles, what they do is, like I said, they increase that droplet size spectrum and they also reduce our driftable line. They do that by less than 10% with these 200 microns. So we know that when we increase our droplet size, we have a tendency to maybe lose some coverage. Larger droplets, though, equal less drift. So the design of the nozzle can ultimately manipulate that drift potential. And then what we're going to be looking at is the increase of that droplet size, like I said, could reduce our coverage and ultimately reduce our herbicide XD. So my overall objectives of this study is to look at the influence of the spray nozzle design and the weed density, which in this case we're going to be talking about food stress, and how they interact with our herbicide coverage and our deposition of the dicamba plus glyphosate post-application. So my materials and methods of trial design, I had a randomized complete block with 2 different factors, and those 2 factors we're going to be talking about a little bit later. And we planted dicamba tolerant soybeans on May 15th, and they were planted into 38 centimeter rows. This is the first factor that I'm going to talk about is the different nozzle types that were used. I used a Turbo T-Jet, a medium-sized droplet, and our Hypro Ultra Low Drift. They produce a very coarse droplet, and then finally the last one is the Turbo T-Jet Induction. They produce an ultra coarse size droplet. Again, the ultra coarse is the biggest droplet size that we can get. The second factor of my study was looking at different weed densities. So on May 16th, we applied a pre-emergent herbicide that was pyroxasulfone. We applied this at different rates so we could establish and manipulate different weed densities within my block. So you can see right here in this chart, we have 6 plants, 14 plants, 17 plants, and 25 plants as my 4 different averages for my density. And these densities were then established in a quarter-year quadrant. We established these quadrants prior to the post-application. That post-application was applied with an ATV traveling at 16 kilometers per hour, and we had a 3-meter side boom that you can see in this photograph. We had a pressure of 140 liters per hectare, and we had a pressure range from 262 to 290 kilopascals. We can see a range here with the pressure just due to which nozzle type is used. This application is to mimic an commercial dicamba application that you're going to see in the field. The application was applied on June 18th, and they were applied to 5 to 10 centimeter size plants. What was in that post-application consisted of 1,100 grams per hectare of glyphosate, 560 grams per hectare of dicamba. We used on target 0.5% volume of volume, and we used Vision Pink dye 0.25% volume of volume, and we had a fluorescent dye mix which was called Spectra Trace SHP. We used Promotocoat spray cars within the soybean canopy, and this was to Vision Pink that I was talking about. They visually mark up those spray cars. Once the spray cars were sprayed with ATV, we took them back into the lab and then we scanned them in 600 by 600 images. Now I did analysis of those spray cards using an APS access software to look at the percentage of coverage and the deposition per centimeter squared. And these cards are also about just the regular size sheet of paper. Here we have a visual of the spray card I was talking about. So you can visually see that our turbo TJet gives us the highest percentage, and that percentage is 21%. We have 39 depositions per centimeter squared. Then we have the hydro low drift nozzle, gives us 17% coverage, 28 depositions per centimeter squared, and we have the turbo TJet induction, gives us 12% coverage and 18 depositions per centimeter squared. The turbo TJet and the turbo TJet induction are completely different when we look at coverage. But the hydro low drift nozzle are somewhat similar to both of them. When we're talking about just depositions per centimeter squared, all three nozzles are different when we look at that. So we know that coverage is important, and it was important to look at the coverage and analyze coverage, but we wanted to know how much spray solution is actually deposited onto our goose grass plants. So immediately after our application, I had a great team. We took 2 samples from each quadrant taken out from the plots. They were washed in a 200 ml solution of surfactant, which was Trident and water. And then we used a leaf area meter to measure those samples that I was talking about, measure their leaf area, and then we took the samples that were rinsed, that solution, we were able to do the raw fluorescence using a thermometer, and then using those 2 numbers of the leaf area and the thermometer, we were able to calculate a standard and then calculate how much spray solution was actually deposited onto our goose grass sample. So here's getting into a little bit of the data I have collected so far. This is just looking at the influence of the spray nozzle design, not regarding our weed density, and we have a range of 1.4 to 1.7 microliters per centimeter squared. There is no difference regarding our nozzle type of how much spray solution was deposited onto our goose grass plant. Here is looking at another chart of just the weed density, not regarding our spray nozzle design. We have a range of 1.2 to 1.7 microliters per centimeter squared, and we do not see a difference versus our 6 plants to our 25 plants of how much spray solution was deposited onto our goose grass plant. Looking at the interaction between both, our spray nozzles are across the top, and our weed density is right here next to me. We have a range of 1 to 2.3 microliters per centimeter squared. You can see there's no difference when you look at that p-value, but the interaction of the weed density and nozzle design on how much spray solution was deposited onto our goose grass plant. So we know that we didn't have any differences when we look at how much spray solution was deposited onto our plants. Then we want to look at maybe, do we see any difference when we talk about the percentage of control we're getting? So we have a range of 92 to 95% of control looking at our goose grass, which was done 21 days after our application. And there is no difference regarding which nozzle was used and how much percentage of control we're getting in 21 days. This is looking at the weed density, again, on regards of how much percent of control we're getting. We have a range of 91 to 98% control, and we do not have a difference on the level of weed density we had, and the percentage of control 21 days after application. This is looking at the interaction between our weed density and the nozzle design. Again, 21 days after application, our percentage control, we had a range of 84 to 99%, and we have the same amount of percentage of control of our goose grass plants 21 days after application regarding our weed density and our nozzle design. So with our conclusions, we can say that the spray coverage was reduced when we use a medium-sized droplet compared to our large droplet. But when we look at how much spray solution was deposited onto our goose grass plants, we can say we provide adequate amount of dicamba plus glyphosate to those plants with our post applications. Weed density did not have an effect on how much spray solution was deposited onto our goose grass plant, and we had an adequate amount of weed control in terms of our weed density and our spray nozzle design 21 days after application for our post application of dicamba plus glyphosate. So when we look at some future research, I had two trials in the field that didn't work out. I guess you could say they failed. I was going to look at Palmer and Waterhead. We weren't able to manipulate those densities that we wanted to. So right now I have an going trial in the greenhouse right now in Princeton, looking at water hemp. And next summer, hopefully, we can figure out how to manipulate those different densities so we can look at water hemp. I'm excited to see if we see any differences on a broadleaf species of a weed versus a grass, if we have any differences there, or we're still going to see the same with our grass species. And at this time, I will take any questions. Yes.
So I guess from your talk, what I heard this morning, dicamba isn't effective on grasses, but just broadleaves. So goosegrass, not a grass, is it a sedge or something else?
No, it's a grass. It's a grass species.
So how does dicamba work on a grass species?
Well, we're just looking at actually how much spray solution was deposited onto those grass species. So I guess I'm not 100% sure that dicamba will be working on that, but we have the dicamba plus glyphosate mixture there. So usually glyphosate more or less takes care of a grass species when we see the terms of control. But like I said, I'm not sure which chemical the interaction between both is taking care of that for that grass species.
Well, we have
Yes.
time. So are you going to recommend ultra coarse nozzles?
In terms of like an ultra coarse, or well, you mean between the high-pro or the heater turbo heat induction, or just one of those in general?
One of those in general, the one that produced the coarsest droplet.
I mean, personally, I would recommend it, but if you're spraying dicamba, you really don't have a choice since you have to use a nozzle that is approved. So this results just give you the fact that when you do use that nozzle, you provide adequate amount of spray solution deposit onto your plant.
Is there any big cost difference between the nozzles?
I'm not sure on the different economics, looking at how much each nozzle costs.
Questions? All right, so next up, moving along, we're going to have Ms. Sumudu Karunadasa, who will be giving us her presentation on cytokinin signaling in rats.
So I'm Sumudu Karunadasa and I'm working in local asthma Islam. Today I'm presenting some of the data about cytokinin signaling and its promoting effect on protein synthesis. So before going into detail, let's see what is cytokinin and what are the functions of cytokinin in plant. Cytokinin is an important plant hormone which regulates plant growth and plant development. And there are many functions of cytokinin, but here I have listed some of the functions. As the name implies, it promotes cell division, and it is also important in the process of delaying premature aging in plants. And cytokinin is also important in medicinal function, organogenesis, and lateral bulk development. How cytokinin regulate all these processes? This is through a signaling cascade, and this figure shows the cytokinin signaling pathway. And cytokinin signaling pathway consists of 4 major components. The first major component is histidine kinase receptors, and there are 3 major histidine kinase receptors known as AHK2, 3, and 4. They are usually located in plasma membrane and the membrane of endoplasmic reticulum. And upon binding cytokinin, the conserved histidine residue gets phosphorylate, and it transfers the phosphate to conserved aspartate residue. And the second major component in cytokinin signaling is histidine phosphotransferase protein. There are 5 members of AHP protein, and they continuously translocate between cytosol and nucleus. So an AHP protein transfers phosphate group to another 2 members in cytokinin signaling. They are type B response regulators and type A response regulators. So type B response regulators activated through phosphorylation and they upregulate cytokinin responsive genes. And they will also upregulate type A response regulators. And type A response regulators act as negative inhibitors of cytokinin response, and it controls both the duration and the strength of cytokinin signaling. So when considered type B response regulators, there are 11 components of type B response regulators. In my study, I'm focusing on ARR1. ARR1 response regulator is known to express in almost all the plant tissues, so it is an important component in cytokinin signaling. So these are the plant lines I used in my research. ARR11 mutant plant line, it is generated by using a tDNA insertion. It contains NPT2 gene as a molecular marker, and this NPT2 gene gives it kanamycin resistance. And the second plant lines are ARR1 overexpression plant lines. They also have been generated in the ARR11 mutant background, and these genes, they are the ARR1 gene has been expressed under the promoter of 35S-CAMP. So the third plant line is phosphomimic ARR1 plant line. They are the conserved aspartate residue at the 94th position has converted into glutamate residue, and it mimics the phosphorylated form of ARR1 protein. So in this plant line, it resembles the activated form of ARR1. And this plant line also has been generated in the ARR11 mutant background. Figure A shows one of the nature observations I got in my research. It shows the rosettes of the seedlings that grow in control and kanamycin media. It shows wild type ARR11 mutant and several ARR1 overexpression lines generated in the ARR11 mutant background. So as expected, the ARR11 mutant line shows a resistance to kanamycin compared to the wild type because it contains the NPT2 gene as the selection marker. And but ARR1 overexpression lines, even though they have been generated in the ARR11 mutant background, it shows some sensitivity compared to the ARR11 mutant. Because there's a difference in these sensitivity levels between ARR1 overexpression lines, we check whether there is any correlation between the ARR1 protein expression and sensitivity level to kanamycin. So figure B shows the protein dot, which shows ARR1 protein levels in these plant lines. Here you can see that ARR11 mutant does not have any ARR1, but ARR1 overexpression lines contains higher ARR1 protein levels compared to the wild type. The member with the highest ARR1 protein shows the highest sensitivity to kanamycin. So we can say there's a correlation between ARR1 protein expression and the sensitivity to kanamycin. And also we check the same thing by using phosphomimetic overexpression plant line. As in ARR1 overexpression line generated in ARR1 background, phosphomimetic lines also show sensitivity to kanamycin. And figure D shows the growth response assay of these plant lines in a range of kanamycin doses. The gray color line shows the ARR11 mutant, which shows resistance to a range of kanamycin doses. But the ARR1 overexpression lines and the phosphomimetic lines, which are denoted in green color and red color, shows less resistance to the same kanamycin doses. And we also tested these plant lines in the presence of another antibiotic, which is spectinomycin. So in the presence of spectinomycin, we got completely opposite results to the kanamycin. And here the ARR1 mutant showed sensitivity to spectinomycin compared to the wild type. But both ARR1 overexpression lines and the phosphomimetic overexpression lines show resistance to spectinomycin. Kanamycin and spectinomycin are aminoglycoside antibiotics. They inhibit protein synthesis in different ways. Kanamycin, in the case of kanamycin, it binds to 30S ribosomal subunit and it misreads the mRNA. And these results misfolded protein and subsequently they result the protein aggregates. In the case of spectinomycin, they bind to 30S ribosomal subunit and it prevents translocation of tRNA. So therefore, spectinomycin completely inhibits the protein synthesis rather than misreading the mRNA. Due to these results, we got for the kanamycin and spectinomycin. We thought these differences could be due to the differences in protein synthesis rates. So to check that, we used furomycin. Furomycin allowed us to detect newly synthesized protein levels in these plant lines. This protein blood shows the furomycin level protein. So as you can see that there's a lower newly synthesized protein levels in the ARR11 mutant compared to the wild type, but there is a higher newly synthesized protein levels in both ARR1 overexpression and phosphomimetic overexpression lines generated in mutant background. And here I also use cyclohexamide, which is an eukaryotic protein synthesis inhibitor, and there's a drastic difference in the samples treated with cyclohexamide and with untreated samples. So from these, we can conclude that increased cytokinin signaling in ARR1 gain of function increases the sensitivity to compounds that transcription, and also from these results you can say cytokinin signaling is important in controlling local protein synthesis rate. In future, we would like to evaluate global protein synthesis rates under different abiotic stress conditions.
How did you quantify protein synthesis rate? Is it just whether or not protein synthesis occurs, or did you have a way to quantify the rate?
We can quantify from the loss, protein loss.
Oh, so it's
Yeah. And this shows the puromycin level protein, and there's a significant difference between these lines.
So antibiotics and plants is studying the response to antibiotics artificial because your plant had the NPP gene, or do plants normally respond to antibiotics if they don't have the transgene?
Usually plants, these are the antibiotics that inhibit protein synthesis in different ways. So in the presence of kanamycin, the plants that have NPP2 gene shows a resistance, but even the, when we assess other plants that do not have NPP2 genes, they could, those plants can show different sensitivity levels. In the plants that have NPP2 genes in the different expression levels, they use different sensitivity levels to kanamycin. So, but the NPP2 gene only shows the resistance to kanamycin. Here I have used pectinomycin, and I have done research with chloramphenicol and all. They show different sensitivity to those antibiotics as well.
Any questions? Thanks,
Oh, you have to say it into the microphone.
And questions?
That
Any questions?
Oh, no question, sorry.
All right. Well, thanks very much. Next up, we have Katherine Rod, who's going to be giving her presentation on soybean crop management.
All right, can you hear me? Perfect. All right, so thank you for the kind introduction, Lane. Today, I'd like to talk to you about one of the projects of my PhD thesis. So here in Kentucky, you are in a unique situation where you can grow 3 crops in 2 years on 1 piece of ground. And so with my PhD research, we're looking at Kentucky's winter wheat and double crop soybean system. And so the project today that I'll be talking about is intensive management practices for those double crop beans. So soybeans are a crop that is photoperiod sensitive, meaning that the day length greatly dictates the growth stage and their reproductive development. And so when you're planting later in the season, the day length is changing rapidly. So we're going to focus our attention to the graph on the left. These graphs represent soybean planting date on grain yield. Studies that have been conducted here at the university looking at this. And so normally when we plant double crop beans, we are planting in this June region. And so especially in West Kentucky with maturity group 4 soybeans, we are only reaching 70 to 80% of the maximum yield that those soybeans could have made if they were planted on time as full season beans. And so planting date is important, and we're also going to look at intensive management practices to help get as much yield as we can in our short growing season. So the objective of this project follows a complementary winter wheat study where we have different harvesting timings that correlate with the different planting timings of our first objective. Our second objective for this study is to look at different intensive management practices in both planting timings and seeing if any of those would help increase yield as well. So a little bit about materials and methods and design setup. This study so far has been planted in 2017 and 2018 at the UK Research Farm in Princeton. on 2 different soil types, a Kreider silt loam and a Zanesville silt loam. So I have 4 failures here that I will be presenting. The soybean cultivar we use is P35T58R. Luckily, we've been able to have it the past 2 years, so it's the same. This is planted in 38 centimeter inch rows into wheat stubble. And so we have 2 different planting timings. Our early is planted at after wheat grain harvest has been harvested at 20 to 22% grain moisture. This is a little early for wheat, and then we were able to get our early planting in. Otherwise, our late planting timing is when wheat is normally harvested at 13 to 15% moisture. And usually there's about a 10 to 14 day difference between the 2. And then within each of those planting timings, we have 2 different seeding rates. We have our low seeding rate of 375,000 seeds per hectare and our high seeding rate at 555,000 seeds per hectare. We also are doing a seed treatment and a non-seed treatment. The treatment we chose was Pioneer Premium Seed Treatment. It includes 2 fungicides, Allegiance and Everglow, and an insecticide of Gaucho. We're also doing a foliar pesticide application at R3. This is either a prophylactic or a blanket application, regardless of disease pressure. And the other treatment is the UK recommendation at economic threshold. So whether you need it or you don't, depending on your insect and disease pressure during the growing season. So a little bit about the stats. We used proc limits and we analyzed both 17 and 18. Year in replication was random factors. And then we used contrast statements to do mean separation. The thing I would like to note about this is for our foliar pesticide applications, I only have 2018 year data. That was the only year that we were also able to scout and apply as needed fungicides or insecticides due to man labor. So the thing to note of what year this data is coming from is over here in the top right corner. Otherwise, we'll look at planting timing first. So with this, we saw that there was a significant difference between our 2 planting timings. And this is something that we would expect because so soybeans planted in West Kentucky after mid-May, they lose 0.5% a yield per day after mid-May. So with this, when we were planting in early to mid-June, with our early planting timing, we're able to capture more of the growing season comparatively to our late planting timing. And for those of us who are not the best at metric measurements, this is about a 12 bushel yield difference. So within each of those planting timings, I have 6 different treatments, and they're, for all intents and purposes, I label them UK, plus, and then super with a higher seeding rate, and then plus. So UK current recommendations are the low seeding rate, no seed treatment, and only foliar pesticide applications as needed. And those foliar pesticide applications are Quadris Top, which is your fungicide, and Warrior 2 is Xeon Technology, which is your insecticide. And then we stack them up as we go, and then with our Super Plus being our most intensive management treatment of the high seeding rate using the seed treatment, and then doing that prophylactic application at R3 regardless of economic pressure from your diseases. So this is all 6 treatments stacked up against each other. And again, this is only for 2018 because of the correct application of the foliar treatment. And so the interesting thing to note is that our Super Plus has was our highest yield, which is it's always nice to see when you put everything on it that it actually responds to it. However, and it is significantly different than our UK recommendations. However, I would like to draw your attention to the UK plus, the super, and the super plus. All of these 3 are not significantly different than the super plus, nor are they significantly different than our UK recommendations. And so when we stack these up against each other, we want to see if any of the main effects also are driving any of these changes. So first, we'll look at seeding rate. So our low seeding rate had a significantly lower yield comparative to our high seeding rate. And again, for those of us not the best at metric, this is about a 5 to 6 bushel difference between the two. And this, the high seeding rate, having the higher yield, makes sense because of the more plants we have per acre, which can very easily be seen in these canopy closure pictures. These are pictures that we take once a week over the canopy. We analyze them using Canopio, and so Canopio analyzes the picture and it accounts for the amount of greenness in the picture, which translates to canopy closure. So over here on our left, we go from early planting with a high seeding rate all the way down to our late planting with our low seeding rate. And these percentages are percent canopy closure or percent greenness of the photo. And so what I like to draw your attention to is even in each planting timing, the late and the early, the high seeding rate always has more greenness or more leaf coverage or canopy closure than the low seeding rate. And this is important, especially with soybean, where we're able then to close the canopy and capture, and the plants are able to use that solar radiation more efficiently and capture more of it to produce and use on grain fill. So our next treatment is seed treatment. And this one kind of surprised us that the seed treatment had a higher yield, a significantly higher yield than our non-treated beans. Usually when you do a seed treatment recommendation for soybean, you recommend it on cool season beans because those beans are being planted in the cool, wet soils where you have favorable conditions for seedling pathogens. However, in Kentucky, in June, it's hot. And it's usually not those type of conditions. And so we were surprised to see this. However, the past 2 years, it has been a little bit more, it has been on average or more precipitation in West Kentucky during the planting timing and early seedling growth. Another thing we noticed this summer, especially with our BC and V1s, who are very, very short and young soybeans, we saw a lot of insect damage on them, especially those in the untreated ones versus the ones that had seed treatment on them. And so we're going to keep track of those for next year as well to see if that actually does have an effect or not. Our R3 application also, our prophylactic or blanket treatment also had a significantly higher yield than our as-needed treatment. And this was also surprising to see because in the extension platform, we only recommend that you apply fungicides or insecticides as they are needed with economic thresholds. However, if you do apply any type of pesticide, hopefully you will see an effect from that. And so we did here. Even though we sprayed 3 out of the 4 fields this year for insecticide, we were able to see from the ratings we did at 14 and 28 days after treatment that the plots that did receive the R3 prophylactic application did have a significantly lower amount of insect leaf defoliation than the other treatments. So for future work, this will be planted again in 2019 as our 3rd year of the study on the 2 locations. So in total, we will have 6 study years. We're going to continue to track early season insect damage, We're also going to continue to scout for economic thresholds for our R3 pesticide application. And then because this is the 3rd and last year of this project, we're also going to look at the economics behind each of these management treatments and how they play a role into the economics of the full winter wheat and double crop system in Kentucky. So with that, I'd like to thank my lab, Dr. Karunadasa, for my co-advising along with Dr. Carl Bradley, and then Connor and Josh, who are technicians out in Princeton. This would not happen without them, along with Ethan and the rest of the interns for my project. And so with that, I will take any questions. Yes, Dr. Egley.
Did you measure stand?
Yes. So the question was, did I measure stand? And yes, I did early stand counts, and then I also did harvest stand counts.
But does the seed treatment affect the stand?
So the other question is, does seed treatment affect the stand? And when I did run the harvest stand counts, it was not significant with my alpha of a 0.05. However, it was at like a 0.1 or 0.2. So it was trending into being significant with the seed treatment with that. Yes.
Did you use a specific camera to screen throughput phenotype like the canopy?
So the question was, did I use a specific camera for the Canopio?
Yeah,
No, I used a normal point and shoot camera. The thing to really be careful about with Canopio is how far that camera is above the canopy will change your perception of the amount of green in your photo that the program will read. And so to do that, we hold the camera at a steady level, and we have an aid that is always making sure that we're at within a small variable.
So the camera was stable at the.
Yes.
Oh.
Yes, the camera was stable. Okay. Go ahead.
You go ahead.
Go ahead.
Which of these treatments do you see to be the most promising?
So the question was, which of these treatments currently do I see being the most promising? It's hard to say right now because the economics have not been done, but if I had to pick one, currently I would pick the seeding rate, just because, especially when we stacked the treatments up against each other, the supers were all not significantly different from each other, even though the super plus plus and the super plus had additional treatments or additional inputs on them. Just the additional seeding rate was still significantly the same.
Still have time?
Yeah.
Okay, so the seed treatment effect was a little surprising that it was significant overall because the UK plus was a little less than UK, and the super plus was no different from the super. So was it a seed treatment effect, or was it an interaction between the seed treatment and the prophylactic fungicides and insecticides?
So the question was concerning about seed treatment and whether seed treatment was affected by the prophylactic application as well. And so to combat that, so for your question, when I looked at all 6 of them together, I looked at just 2018 data. However, when I presented the main effects, that's both 17 and 18. But we also ran contrast statements, so then we could go ahead and pull apart just the non-treated scenes, so the controlled versus the 2 treatments that had just seed treatment on them to see if those were significantly different, and disregarded or did not use in that analysis the treatments that have the prophylactic application. So we were able to see that there actually was a significant difference between the treated beans and the untreated beans. All right, thank you.
All right, and lastly, we have Mohammad Dawood, who's going to be giving his presentation on spider mite repellency in tomatoes.
You put the microphone on, please.
Good afternoon, everyone. Thank you for coming. Okay. So our journey with this experiment started in 2017 when we looked at 7-FP zingiberine in white tomato. And then what we got is 3 major allelochemicals component in wild relatives, especially Solanum abocatus. So the title of the research is spider mite repellency or 7-zingiberine, 9-hydroxyzingiberine, and 9-hydroxy-10-11-epoxyzingiberine that isolated from wild tomato. Okay. So tomato Solanum lycopersicum. What Solanum lycopersicum means heirloom varieties or local varieties or cultivated, whatever, so local varieties. So it's one of the most economically important in the world. However, tomato is a host of several types of arthropods that reduce productivity, especially spider mites, aphids, whitefly. We know tomato breeders have focused on increasing fruit quality and quantity, but let's focus on arthropods. Because of these pathogens and disease, spider in tomato, in most cases, considered a high-risk crop. Why? Because it needs intensive amount of pesticides, insecticides, and so on. We can see that in this picture. So we know in Solanum lycopersicum, there is an activity, there is a resistance. But this resistance is not much to decrease this pesticide and insecticide. What's the solution in this case? Well, solution we have, wild type of tomato. Here in this picture, we have Solanum lycopersicum by the scientist group as a resistant for arthropods, especially for whitefly, aphid, and termites, spider mite, because of natural compounds that included in the secretion of this type. Okay, so it's good for health. Yes, good for human beings because we will decrease the amount of pesticide, and then the tomato is going to be more resistant. The objectives: does other allelochemicals besides 7-zingiberine? By the way, we have zingiberine, we have 7-zingiberine. Zingiberine came from ginger, it's normal ginger. 7 came from wild tomato. Yeah. So here, okay. So we can see in this picture, so spider mite, and then we have eggs of spider mite. The picture of spider mite. Plant materials, I used LA2329, 2 excisions A and B in 2017 and 2018. This is the image from GCFID for LA2329. Why I chose this? Because of three major allelochemicals, which we already tested zingiberine. So like rotation time, we see the rotation time at 5.7. And we want to know the hydroxyzingiberine and epoxy. So let's test them. And then if they have activity, or why not to transfer them from wild to cultivated? Okay. What I did, I separated. I used the silica gel to separate 3 major allelochemicals. See the black spot here? The black spot. Here, the first one. Black spot here in this is zingiberine. And then the second one, 18, 17 is tumor. And from 32 to 40 to 39, that's hydroxy. And then the last one, say we have 4 groups I separated successfully. And then, by the way, when I separated, the purity of zingiberine was 98%. And then epoxy, 86%. And then for the hydroxy also 86%. This is the steps that I used. Okay, I used 2 machines. First, I used GC gas chromatography to know how many allelochemicals component do we have in this wild tomato, and then after that, I needed to know what is it. I needed to know ID of this chemical component. That's why I used GCMS. Gas chromatography and mass spectrometry. Okay. This is I use the MDES32 software. That's this image data taken from GCMS, and then I put it for the MDES to know ionization of at 220. This is the in 220. I got it, the hydroxy. This is just one sample. So I have more, but this is just one sample for the weaken of the hydroxy, C15, H24O. So on epoxy, we have O2, and then if we remove oxygen here, it's going to be 7-ethylzingibrine. C15, we know the sesquiterpene monocycle hydrocarbon. Yeah. In my journey, I used this bioassay. This bioassay proved by Dr. Schneider in 1993. The idea is we have arenas, and then we have a C and T. C means control. I use just hexane. Then for the T, I use just treatment. Treatment by known level of concentrations from 3 major components. Like individually, I use them. I use 30 mics, spider mics. That's the rare and broad in this bean Fasciculus vulgaris. And then what I did, I put the spider mite, I sent it right in the middle of the bridge. You see the bridge. And then I washed in which way the spider mite should go. If the spider mite goes with control, that tells me this chemical has activity. If the spider mite goes through the treatment, that indicates this treatment has no activity. And then we can see here, you can see that I have a small video here. See the spider mite here? This is treatment, and then this is control. It moved away from the treatment. Okay, in that I use probe it in SAS, and after homogenize or so exit ratios to know EC50, and then after that, after homogenization between the bridges, 2 bridges, I use the probe it. In 2017, like I said, we had 2 groups. This group had just in this chemical secretion or trichemical secretion had just 1 major endochemical, which is 7-ethylzingibrine. And then this one they had 2 major endochemicals. When I tested based on the zingibrine area unit, microgram per centimeter square, at the low concentration here, so at this level also we got activity, but here at the low concentration, here maybe close to the 0, and we got activity. We couldn't know what's going on in these materials. When we go back to GC and then GC-MS, we got 2 major endochemicals. By the way, here we got just epoxy, but after analysis, we got there are another endochemicals called epoxy has activity. That's why we started to separate between 3 major endochemicals. Like I said, the purity of the epoxy was 98%, and then for hydroxy 86%, and then for epoxy 87%. This is the fractions. Okay, so here in EC50, we saw really there is a significant difference between epoxy and hydroxy zingibrine. So there were no differences between hydroxy and epoxy. Okay, we can see the differences between zingibrine and hydroxy and epoxy, which is 10-fold difference. So this is just type model that I put it here to know that our model is work very well. So at the log, we can see the EC50 here, EC50. Okay, so EC50 for this one, just go this 0.0013. This is a low concentration. 50% of the population, spider mite population, repealed by this amount of microgram per centimeter square. In conclusion, we knew that 3 major endochemicals had activity against Tetranychus urtica, but the activity of hydroxyzingibrine and epoxyzingibrine were like huge active compared to the zingibrine. Any questions?
Is there any attempt to breed tomato plants that have higher levels of active components?
That's our goal. So in my presentation, just I focused in chemicals, organic chemistry. Our goal is to breed. Okay, we got it. We got some successful in the field this summer. We had genotypes. Okay, had activity. At the same time, we're not focusing on these chemicals. We have different chemicals. What we're doing, we're transferring phytochemicals. Okay, not just the organic chemistries we're focusing on tomato secretion. No, there are phytochemicals we're focusing on phenols or flavonoids. Okay, we know that by increasing the phenols of phenols, we will eliminate different diseases. Just yeah, we have some success.
Is there any idea why spider mites don't like these?
Well, that's a good question. So the structure, maybe the sensitivity of the spider, so the chemicals, so the spider may be like toxic for them, maybe because of the so the circle here, we know the two double bonds with the hydrogen, and then maybe oxygen, we have an epoxy, or oxygen with hydroxy, which is like 3 major components attract.
Do they? Is there evidence that they suffer damage, or are they just
don't like it? If we remove these 3 major radiochemicals, okay, maybe they like it. If we put it, I don't know, they will die or not. But my colleagues worked on it in some point, yeah.
Do you have any concern with it repelling beneficial insects? Do they fall on?
Yeah, specific?
Like, I don't know, pollinators, bees?
No. I think so, just for arthropods. Because there is a good relationship between bees and the tomato. So, good relationship. That's why I put the bees, the structure on top of the tomato. So, it's good pollinators, especially or actually bumblebees, not honeybees, bumblebees. So, good friend. Yeah, I love it. Any other question?
I have one more question. Do you know the biosynthesis pathway for these?
Yeah, yes, good question. So for biosynthesis, for the zingerine, so the second and third major radiochemicals came from the same biosynthesis of zingerine. So there's a guy called actually Bleeker. Bleeker worked on it, and then finally 2 years ago, one guy got patent on it. Actually, Dr. Snyder started to work on it, but we a little bit late to get it. Someone got it. So, well, but they didn't got all of them. We still will focus on specific pathways. So.